Good morning, everyone. This is Dipanwita from the Department of Microbiology, SURENDRANATH College. I am presenting a review paper on analysis of microbial biodiversity by metagenomic study in the Mangrove Sediment of Sundarban, West Bengal. Focusing on the theme Microbial Biodiversity and Sustainable Development on Human Welfare, metagenomic study is of utmost importance. Microbes, despite their small size, have a huge impact on our lives. Microbial world, a treasure in itself, if exploited judiciously, can contribute to the sustainable development. Soil being the living entity of the earth and the main reservoir of microbial biodiversity, studying soil microbial biodiversity is very much important. Microbiological studies in the soil environment are hampered by the fact that the largest proportion of the soil bacteria as yet cannot be cultured, and the microbes found using common culture methods are rarely abundant in any environmental niche from which they are cultured. This suggests that there is the need of molecular techniques which overcomes the need to isolate and study each organism for in-depth characterization. Now this condition leads to the path of metagenomics. Now the question arrives, what is metagenomics? It is the study of genetic material recovered directly from environmental samples. It provides a new way of examining the microbial world that not only will transform modern microbiology but has the potential to revolutionize understanding of the entire living world and their functions in different ecosystems. In metagenomics, the power of genomic analysis is applied to entire communities of microbes, bypassing the need to isolate and culture individual bacterial community members in order to identify the microbes in the community. Metagenomics combines the power of genomics, bioinformatics, and systems biology. Now comes why is metagenomics important. As I have already said before that 99% of the microbes are not accessible to us, so we need a culture independent technique to overcome this problem. That's why metagenomics is of utmost importance. Moreover, it can lead to the discovery of many novel natural products, new molecules, enzymes, etc. Also, we can come to know about how do microbial communities work and how stable are they. Now we have to understand how all metagenomic studies take place. At first, the genetic material DNA can be extracted from any samples taken from the environment, such as described in the picture here. DNA is extracted from soil, seawater, and human gut. Then it is filtered in the next process. After that, it is multiplied by cloning, and then it is sequenced. These short sequences are then put together again using assembly methods to deduce the individual genomes or parts of genomes that constitute the original environmental sample. Now, mainly two types of metagenomic sequencing is used to study microbiome. One is the 16S rRNA or targeted sequencing, which utilizes PCR to target and amplify portions of hypervariable regions of bacterial 16S rRNA gene. Amplicons from separate samples are then given molecular barcodes, pulled together and sequenced. After sequencing, raw data is analyzed with a bioinformatics pipeline which includes trimming, error correction and comparison to a 16S reference database. After the reads are assigned to a phylogenetic rank, a taxonomy profile can be generated. But in shotgun metagenomic sequencing, all given genomic DNA from a sample can be sequenced, unlike 16S sequencing, which only targets 16S rRNA genes. The library preparation workflow is similar to regular whole genome sequencing, including random fragmentation and adapter ligation. Because shotgun metagenomic sequencing covers all genetic information in a sample, the data can be used for additional analysis, for example, metagenomic assembly and binding, metabolic function profiling, and antibiotic resistance gene profiling. Now I am going to describe 3 research articles based on this metagenomic study in the Mangrove Sediment of Sundarban. Now, why the place chosen is Sundarban? Because Sundarban is the world's largest coastal sediment comprising of mangrove forest and provides a unique ecological dynamics for diverse microbial community that play an important role to sustain productivity, conservation, remediation, and the overall sustainability of the ecosystem. Now the first paper which I am describing here is the culture-independent molecular analysis of bacterial communities in the mangrove sediment of Sundarban, India. This research was conducted by Obhrajyoti Ghosh and his team and it was published in 2010. They collected soil samples from top 15 centimeter of 5 different sites on the islands of Sundarban. 216S rRNA genes libraries were constructed and partial sequencing of the selected clones was carried out to identify bacterial strains present in the sediment. Phylogenetic analysis of partially sequenced 16S rRNA gene sequences revealed the diversity of bacterial strains in the Sundarban sediment. At least 8 different bacterial phyla were detected. The major divisions of the detected bacterial phyla were the alpha, beta, gamma and delta proteobacteria, flexibacteria, actinobacteria, acidobacteria, chlorophrexi, firmicutes and planktomycetes. But gamma proteobacteria was found to be the most abundant bacterial group in the Sundarban sediment. Next paper is Cataloging the Bacterial Diversity of the Sundarban's Mangrove, India in the Light of Metagenomics. This research was conducted by Piyush Boshak and his team and was published in 2015. In this study, they have reported the profile of bacterial community at variable depth of soil sediment in the world's largest tropical mangrove sediments of Sundarbans, India, using 16S rRNA gene amplicon sequencing. Sediments were collected from 2 sampling stations, Bonicamp and Dhachi. Metagenomic DNA was extracted from soil samples using AMO-Bio DNA Power Soil Kit. To analyze the bacterial biodiversity, the V1, V3 regions of bacterial 16S rRNA gene were amplified by PCR. Pyrosequencing was performed for 200 cycles and the output was 61,363 sequences with size 30 mega base pairs and with a content of 56 guanine and cytosine. In both the stations, Proteobacteria and firmicutes formed the most abundant bacterial phyla with percentages varying at different depths. We can see in the figures bacterial community structure of soil sediment samples from different depths of mangrove forest. One is from Dhachi, 5 meter depth, and the other 2 from the Bonicamp, one of which is from 5 meter depth and another one from 9 meter depth. In this way, their pyrosequencing data revealed the presence of diverse bacterial population in the Sundarban mangrove forest. In the next paper, which was also published in the same year 2015, is about the diversity and distribution of archaea in the mangrove sediment of Sundarbans. Obhrajyoti Ghosh and his team this time analyzed the diversity and distribution of archaea using the same 16S rRNA gene amplicon sequencing. They have collected sediments from 3 distinct mangrove areas, Kotkhali, Bonicamp and Dhulibashani. They have analyzed archaeal 16S rRNA gene amplicons prepared from 6 sediment samples originated from 3 sampling stations at 2 depths. After quality filtering, deionizing and removal of potential chimeric sequences of pyrosequencing dataset resulted in recovery of 1,41,816 sequences with an average length of ranging between 500 to 510 base pairs. They were able to assign 1,39,953 sequences to the domain archaea and to classify all the sequences to the below domain level using BLAST-N in Silva database and Megan. All these are mentioned in this figure. Their taxonomic analysis revealed the dominance of phyla Euryarchaeota and Thaumarchaeota. The distribution of different archaeal taxa and respective statistical analysis revealed a clear community shift along the sampling stations. In the next picture, comparative taxonomic profile composition for each sample is given as a partition of a plane highlighting the 6 major phyla, namely Thaumarchaeota, Methanobacteriaceae, Thermoplasmata, Methanococcoides, Methanolobus and Methanosarcina. Now, what are the future prospects of metagenomics? Metagenomics is coming of age but still gaining momentum. This coincides with rapid improvement of sequencing technologies and with the realization that next bottleneck in metagenomics will not be the sequence data production but computation and data storage. From the experience of the past 5 years, the power of metagenomics is obvious while its potential is still waiting to be fully realized. It is now quite clear that even communities of limited complexity pose major challenges in terms of genomic exploration, highlighting the necessity of much deeper sampling and the need for spatial assembly and analysis tools. Such improvements are possible and imminent given the fast progress in these areas. However, the cost of metagenomic sequencing to high coverage, even when employing the next generation technologies, remains a major challenge. Metagenomic datasets are increasingly becoming more complex and comprehensive, and in silico gene prediction on metagenomic sequence datasets is rocketing. After 2005, enormous information about novel genes, open reading frames, operands from diverse environments has accumulated. Now there is a strong need to focus more on validating these novel genes of metagenomic origin by putting them in action in real wet lab conditions to search for more novel enzymes and bioactivities for bioprospecting metagenomes. Else, we may end up putting all efforts for novel genes in dry lab conditions only. At last, I want to thank the organizing committee of BIJOY KRISHNA Girls College, HOWRAH for organizing such a grand webinar and giving me the opportunity to present my review paper. I want to acknowledge my professor Shri BISWAJIT SAHA for his extreme guidance and support. Thank you.